for the organizers. It's been a really fun time at this conference so far. I'm also coming from the biosensor side of things, although we heard a lot of talks about the fundamental mechanisms of aptamer conformational change today and yesterday. And I will show you how we really harness these changes to be able to enable biosensing of small molecules. And so I will, what happened to my, sorry, I'm trying to get to the next slide and it is not letting me. Sorry about this.
What about the mouse key or the side arrows on your keyboard?
Sorry, 1 second. I'm going to try and reshare. Sorry about that.
That's okay.
Okay, let's see if this works now. Okay, I'm not going to touch anything now. Okay, so I wanted to start out by motivating what we are trying to do is to really understand the complex brain. And as everyone knows, we have billions of neurons in the brain, and I like to equate that to the number of stars in our galaxy. But what's really complicated is that all of these billions of neurons are interconnected, they communicate with one another in highly complex ways. And something that I wanted to show you at the start is that this communication happens through two pathways. The first is electrical, where an electrical impulse from one neuron then stimulates the release of chemicals in the synapse, which is the gap between the two neurons, which are then captured by the receptors of the next neuron, which then triggers the next electrical potential. And so as you can see, it's this electrical and chemical interactions that enable chemical communication in the brain. And I want to show you this because we actually use aptamers as the artificial receptors that can capture the molecules of interest, and then we transduce that as an electronic signal. So we're basically mimicking how the brain actually communicates. And when we think about how electrical recordings can be measured, it's actually very well established. We have methods that allow in vivo recordings. There are flexible biocompatible electrodes that can be placed inside of the brain. But what we do in our group at the Laboratory of Biosensors and Bioelectronics is we work with in vitro neural networks. The reason for this is that we want to simplify the high complexity, because if you have a billion of neurons, it's really difficult to be able to understand how the communication happens at a smaller level. So we are able to pattern single to a few neurons on the surface of microelectrode arrays. So these are just gold or platinum electrodes that can stimulate the neurons electrically while also recording from them. But when I started my postdoc at the LBB, my goal was to be able to add the chemical functionality to this sensing so we can simultaneously record chemical and electrical signaling to get the full picture of the communication. But as you all probably know, it's really challenging when it comes to differentiating similarly structured chemicals. And what we want to sense was dopamine and serotonin as two key neurotransmitters. However, if we look at the biosynthetic pathway, you can see that there are precursors, there's metabolites, there's a lot of different molecules that have the same chemical signatures. And so, of course, aptamers were the selective motifs that enable us to differentiate these similarly structured molecules. And during my PhD work, we isolated a serotonin and dopamine specific aptamer that we validated were able to differentiate these similarly structured molecules. In particular, I want to draw your attention to the fact that the dopamine aptamer was able to differentiate norepinephrine that differs by a single hydroxyl group, which was a really big challenge before. And while today's talk is not about field effect transistors, which was the biosensor that we used, what I wanted to emphasize here is that what we utilize is the conformational rearrangement upon the aptamer binding serotonin or dopamine. And the aptamer undergoes a rearrangement of the negatively charged backbone, which modulates the electric field in close proximity to the surface of the sensor. And so you can either get electrons that are brought closer to the surface when negative charge moves away, or on the other hand, you can get electrostatic repulsion if more negative charge is brought closer to the surface. And what's really nice about this technique is that it does not require redox reporters, dyes, fluorescent markers. It really just relies on the conformational rearrangement of the aptamer itself. And so using this knowledge, when I started my postdoc, I wanted to figure out how I can use this mechanism to do chemical sensing from neurons. But as I showed you before, the neuronal communication happens in a very small gap, in approximately 20 nanometers in the synapse. And so we also have to build sensors that approach this spatial resolution. And so I work with quartz nanopipettes that we build in-house in our lab, and we can create these nanopipettes to have orifices of approximately 10 nanometers. And we can actually tune this opening size from anywhere from 10 nanometers to micron scale. And so the vision was to be able to use these nanoscale pores and use aptamers to gate the current. And so to show you what the setup is, we have a silver chloride reference electrode in the nanopipette, as well as in the bulk solution. And what we physically measure is the current that goes through the nanoscale pore upon applying the bias. And so now if we zoom into this pore, the goal was to couple aptamers on the inside so that we can modulate the opening and closing of this pore based upon conformational change. And what's really unique about these nanopipettes is that they show nonlinearity in their current response. They deviate from traditional electronic sensors in that applying a linear voltage does not result in a linear current, but a nonlinear current. And this is due to the fact that it's sensitive to the surface charge that is confined within the nanoscale orifice, and therefore we are also sensitive to different charge rearrangements that are happening at the surface. And so what we wanted to see was that upon target binding, the aptamer conformational change would gate the flux of ions going through the pore, and we would see a change in the current response. So I'm actually trained as a surface chemist. So the first goal was to be able to put the aptamers inside of these nanopipettes. And so we assembled aminosilanes on the surface of the quartz. And then with coupling chemistry, we would couple the amine on the surface of the quartz with thiolated aptamers. And then these pipettes were ready to conduct sensing. But also what's really nice about this system is that we have some quality control in that we can use this current rectification, the nonlinear behavior, to see what we have at the surface. So if we start out by having a bare pipette that has a few negative charges, you see that it's a relatively linear slope. Upon assembling the aminosilanes that are positively charged, you see this inverse rectification, where now we see a positive rectification. And finally, with the highly negatively charged aptamers, we see another inversion and a highly curved current, which shows that we have a lot of negative charge, and this is quantifiable. And so once we validated that the surface chemistry was working, we could start with the sensing. And what was more, we can sense in complex environments with these pipettes. We are measuring in neurobasal media in which neurons are traditionally cultured. So this involves 20 amino acids, growth factors, lots of non-specific proteins. But as you can see, we can see a clear increase of positive bias with increasing concentrations of serotonin. We can show this as a normalized current response. And as you can see, we have a sensitive regime in the picomolar nanomolar regime. We also validate the sensors by using a scrambled sequence, which has the same exact nucleotides as the original sequence, but we scramble the order to alter the recognition properties. We also demonstrate the selectivity against similarly structured molecules, as well as all of the other non-specific molecules in the environment. And here is a typical current response that I can show you, where if we add the wrong targets, such as L5-hydroxytryptophan or 5-HIAA, we see minimal perturbations in the current. You can just see that you just see the noise. However, upon addition of the serotonin, you really see a clear increase in the current response. And this is something that we can monitor in real time, and we are only limited by the temporal dynamics of the aptamer binding. And so this is really exciting, and we wanted to really start to sense from real neurons. But we came across a challenge in that serotonergic neurons reside in the very deep brainstem of the raphe nuclei. And further, less than 1% of neurons in your brain actually release serotonin. And so we started a collaboration with the Salk Institute that creates serotonergic neurons from human skin cells or fibroblasts, where they reprogram these skin cells to induce pluripotent stem cells, which can then be differentiated into serotonin-specific neurons. And this was a very fruitful collaboration because actually they were looking for tools that could quantify serotonin accurately from their in vitro neural networks. They were using conventional ELISA, which uses antibodies that are specific to serotonin, but they have shown cross-reactivity. And as you can see, based on the standard curve, the concentration released from the serotonergic neurons were really at the detection limit of this technique. And so together with the Salk Institute, what we wanted to do was measure in the media collected from serotonin releasing neurons, as well as a control, which is iPSC-derived astrocytes. These are not serotonin specific cells. And if I show you a very simplified diagram, this is the serotonergic system, and that serotonin is released from one neuron and either reuptaken by a serotonin transporter protein or then captured on the next receptor. So there's two different pathways in which the neurochemical can go. If we now stimulate these neurons with high salt, such as potassium chloride, this actually depolarizes the neuron and releases all of the serotonin stock into the extracellular space. We show with our nanopipette measurements that when we have non-serotonergic cells, we were unable to detect any serotonin. However, for the serotonin-specific cells, we detected approximately 1 nanomolar of serotonin released. And upon salt stimulation, we saw a threefold increase in this concentration to 3 nanomolar. Another system we tested was an antidepressant, citalopram, which now blocks the serotonin reuptake. And so now we also expected an increase in the serotonin in the extracellular space, which is what we saw. But now you can see it as a twofold increase, so not as much as the depolarization. And what was really interesting for the neuroscientists was that the acute treatment of 2 hours versus long-term treatment of 24 hours showed the same amount of release, regardless of the amount of time. So what was exciting here is that we can really differentiate physiological differences from these neuronal systems. And as you can see, the differences are on the scales of 1 versus 2 versus 3 nanomolar, which was something that they couldn't do with the conventional techniques that exist. And so this is really exciting for us, especially because this technique is generalizable. Right now, we are optimizing a dopamine aptamer specific nanopipette to also be able to tackle dopamine releasing neurons. And we have all of the key players in place in our group. We have the microelectrode arrays for the electrical recordings. We have the capacity to pattern neurons, including induced pluripotent stem cells. And we also have a scanning ion conductance microscope that allows us to use these nanopipettes as probes and move in the XYZ direction with nanoscale precision to be able to map the chemical release in tandem with the electrical signals. And now I'm really coming to the end of my talk, but I wanted to include 2 last slides based on all the different talks I've seen so far. And it's been a big challenge for me ever since my PhD to really understand what is actually happening when we undergo this aptamer conformational change and how are we actually measuring these signals. So there's so many different techniques for us to understand a little bit of what's happening, such as fluorescence-based methods, such as FRET, where you put a donor and acceptor fluorophore in specific locations, surface-based techniques such as surface-enhanced Raman spectroscopy, where you assemble the aptamers on the surface of plasmonic substrates, and the vibrational modes of the molecule can be tracked as a function of target capture. I also worked with quartz crystal microbalance, which allows you to see the change in the height of the aptamer, even if it's on the scale of a few nanometers, based upon conformational rearrangement. Circular dichroism spectroscopy tells you what kind of secondary structural motifs exist. And finally, impedance spectroscopy can show you the penetrability of redox molecules to the surface of an electrode based upon the aptamer adopting a conformation at the surface. So as you can see, we have a lot of solution-based techniques as well as surface-based techniques that all give you a small piece of the puzzle, but it just doesn't seem satisfactory in terms of understanding what is precisely happening at the surface of our sensors. And so something I've been working on for the past 1.5 years is on trying to really understand what's happening by visualizing these interactions. And so we have conducted solution NMR of the serotonin and dopamine aptamers. And as an example, I show you here that if we have the serotonin aptamer alone, which is the black curve, upon addition of the target molecule, we see all of these new peaks appear, which is the formation of the G quadruplex structure. However, the challenge with NMR for me has been that these aptamers only adopt specific conformations at high salt. And NMR really fails when you put in too much salt. And so it has been a really big challenge in trying to retain the conformation of the aptamers while getting enough resolution, even in the 2D space. And so we've also been conducting X-ray crystallography, where after, I think, 20,000 screens, we were able to get an aptamer target crystal that we were able to fish, take to the synchrotron source, and bombard with X-rays to get the three-dimensional structure. And currently, we are at a point where we have 4 angstrom resolution for the diffraction pattern, and we need at least 3 to 3.5 to really resolve the three-dimensional structure. So as you can see, small molecule aptamer structural understanding is really a challenge. And if anyone in the audience is really interested in this work or has expertise, I would really love to hear about it, since it's been quite a journey for the past few years. But at this point, I'd like to finish my talk. Thank you so much for your attention, but I'd also really like to thank my group at the Laboratory of Biosensors and Bioelectronics. I also want to thank the students that I've supervised on this work, as well as the Salk Institute, and I'm open to any questions you may have, and please contact me also if you have any questions. Thank you so much.